So thank you for the introduction, and I would like to introduce you to the field of carbon nanodots and their use in bioimaging. So this work was predominantly made by Dominika and Sebastian, who are my PhD students. So why at all carbon nanodots? It's most of all they are very cost-effective fabrication routes. They can be made out of low cost and commercially available precursors such as urea, citric acid, ascorbic acid, or trans-aconic acid, to name just a few. And they can be made with low energy synthesis methods, which these days it's very important. They also show excellent biocompatibility with the long-term colloidal stability, dispersibility in aqueous or other media without any post-synthetic modification. And as can be seen here down, they also show non-toxicity as the cell viability is really high. First, we focused our interest on a tunable synthesis so that we could actually synthesize nanoparticles, carbon nanodots of different colors depending on their actually size. So they show the strong and tunable one and multiphoton excited fluorescence, and as can be seen here, they are very resistant to the photobleaching. So they seem to be really nice materials for further exploring. In the studies, as it was also said in the previous lecture, there is a variety of synthesis methods. We could use either top-down or the bottom-up methods, and among them, for the top-down, it could contain of arc discharge, laser ablation, ultrasonic synthesis, or chemical oxidation, whereas in bottom-up, which is more the synthetic approach, we could use various stimuli such as microwave or thermal deposition, hydrothermal treatment, or plasma treatment. How do we define such structures? They differ in a way of organization of carbon atoms within its structure. So we can have highly organized graphitic-like structures where they composed mostly of graphene, or the carbon nanodots, where we have just the pallets of carbons interconnected with each other, or just completely amorphous carbon polymer dots, where the chains of polymer carbons are structured within a small-sized, bulky material. So we call them the zero-dimensional nanomaterials with strong fluorescent properties. And as I said, the four major subclasses can be easily distinguished. The difference between the carbon, so-called quantum dots, and carbon polymeric dots can be seen also within its characterization by, for example, intensity fluorescence. And above of the synthetic methods, we can also dope those structures with heteroatoms, such as gadolinium, for example, to have multi-purpose nanoparticles. So they may be not only fluorescent, but also magnetic, or we can enhance those fluorescent properties by doping with boron or nitrogen or with the fluor or other materials. So in general, the synthetic routes contains the precursor and then internal and external design of such nanostructures, depending on the chemical and photophysical properties we are interested in. So as I mentioned, our most of our interest was actually on the excellent biocompatibility of such materials, as carbon is, of course, one of the material building us up. So they show long-term colloidal stability because basically they do not have ligands on site. Despite it, they can be dispersed in aqueous media, and as I said, they do not show the cytotoxic effect. Moreover, they can be also functionalized because of the energy bonds between the carbon and other atoms. Remarkable optical properties can be seen the pictures taken in our lab. So they have, as you can see with the naked eye, they show a strong luminescence, and depending on the size, we could see different colors of this luminescence. For the biological applications, we've chosen the interactions with the proteins, and we envisaged it as a new bioimaging markers, eventually as a drug delivery systems, which could be used for biosensing. The question arose, can we use them in multiphoton imaging, and what would be this influence of the presence of this carbon nanodots on the biological structures? For this purpose, we've chosen myelin. So we wanted to dope the myelin structure with carbon nanodots to check its behavior in the biological structure. And why myelin? It's because, first of all, it's a crucial component of the nerve structure. It exhibits a liquid crystal arrangement, which was already also introduced today by Małgosia. And the analysis of this structure raised attention because of its role in the transversion of the impulse nerves. When it's broken, it doesn't work well. As you can see here in the cartoon, the signal cannot be transferred through the neurons, for example. And the studies of the formation of such a multi-layer structures is always interesting, because may also be used for industrial applications. So myelin, as I mentioned, is covering the nerve structure, and when we have the demyelination in progress, it causes multiple sclerosis, but also other syndromes, other neurological diseases. And from our previous studies, we've noticed that doping myelin with gold nanoparticles actually stabilized its liquid crystallinity. So we asked the question if doping with the carbon nanodots will also serve as a kind of a scaffold for this multi-layer liquid crystal structure. The myelin is actually built of the stacked lipid bilayers with the water layer in between them, and the myelin tube consists of many of such layers. Those particles consist of course, of the hydrophobic and the hydrophilic part, and the question was also where eventually our carbon nanodots will go. Will be stacked in between the water layer, or they could go in between the lipid bilayers eventually. So the myelin, first we've observed the myelin growth, and so you can do it under the microscope and observe how the myelin tubes are rising from the phospholipid stack, and then how different structures can be achieved, including also the 3D structures, which may be solid, which could be hollow inside, which can be also looped. This is very interesting, self-assembly of this myelin structure, or oval with loads of space inside. The 3D structures also looked nice, so it looks exactly like a spoon-like, or it could have a bowl-like structure. It's also were wondering if it would have an impact on the organization of the carbon nanodots in such structures. Some time ago, we also doped the myelin with the silicon dioxide nanoparticles. As you can see, they were mostly stuck on the tubes. They never went into the tubes. And actually, I had this correlation with the octopus. So we had yesterday night octopus salad, right? So it looked mostly as this kind of structure. But the question was where the carbon nanodots will go. So first, the temperature, solvent, time dependence give us various not only colors of these carbon nanodots, but also, as you can see, various sizes. I liked it a lot as they have a triangular shape, which is also not a common shape for the synthesis of nanoparticles. So first, heteroatom-free carbon nanodots were fabricated by the solvothermal treatment in ethanol for 2 hours, and we got this cyan-like nanodots for by 9 hours synthesis, where we got the green-like nanoparticles. We defined where we have the vibrational bonds in the polycyclic aromatic hydrobonds and what is the internal structure. And indeed, what we got were the triangular nanoparticles. We got some remarkable linear optical performances of those nanodots. So ultra narrow emission of about 35 nanometers with no excitation dependent tendency and a long-term stability, with the quantum yield of almost 50%, which is also quite remarkable, and the nanosecond fluorescence lifetime. And as seen before, also the sizes actually are decisive about the color of the luminescence. So first, these ones are mostly 2 nanometers, and here we have 2.5 nanometers big nanoparticles. So the triangular nanodots were further scrutinized, and first we focused our attention on the green ones, and we're looking at the doping of the phospholipid-based microstructures with those carbon nanodots. As you can see, most of the carbon nanodots went actually inside of the liposomes, but also we've seen them along of the phospholipids creating myelin. And we're also wondering if the carbon nanodots will perturbate the myelin organization. As you can see in comparison of these two photos, they actually did not, probably because of their very small sizes. And the hollow part of the myelin was also maintained, as well as the organization of the 3D structure. So the spoon-like structure was also achievable even upon doping with the carbon nanoparticles. The oval shapes were also seen, as well as with the myelin structures doped with Nil red. This is a molecule used typically for the studies of myelin. And if you look at those two pictures, they actually do not differ much. It's just different microscope, actually. So as I said a few minutes ago, the question arose if we can use those structures for the multiphoton imaging. Why? Because with the 2-photon excitation, we can get better resolution. It's less damaging for the imaging in general, as it produces less photobleaching, as at once we only excite the femtoliter volumes in comparison to the overall light going through whole the sample. The image is also more focalized on the sample, which is important, especially if we deal with the 3D structure. So for this, we use the polarization sensitive two-photon fluorescent microscopy, where we cannot only image the sample, but also we can follow the direction of the dipoles in the sample, and we get the information about its orientation of the emitting dipoles. For it, we use the polarized light for the excitation of the sample, but also we extract the polarization information from the luminescent material which we are studying. So first, we also checked the two-photon absorption cross-section. If at all, we can actually excite the carbon nanodots, as it's not obvious. It doesn't apply to all the materials. And we checked the laser intensity versus laser power relations. Here we excited with 880 nanometers. And you could see here that indeed it's a two-photon process, both for cyan and for the green nanoparticles, and actually the spectra is pretty complex. And although it doesn't show much of the Gepard-Meyers divided by the Daltons, so the size of the nanoparticles, still it actually exceeded the typical fluorescent dyes used for staining of the biological structures, and with the excitation wavelength ranging from 700 to 900 nanometers, we actually match well the first biological window. These are maps made in metal, but nanoparticles were also luminescent in water. So we checked with the two-photon fluorescent microscopy if we can decipher the inner structure of myelin while using the two-photon excitation. And indeed, we could go more in depth of such a structure by changing the Z position of the myelin tubes in our under the microscope, and also we confirmed that it's a two-photon. Its excited emission is the two-photon process. So we checked it also for the blue triangular carbon nanodots, and here what we've seen was, of course, similar results for the for the 3D structuring, but more interestingly, we've seen also the polarization dependence of the emission. So depending on the light incident laser beam polarization, we've seen different intensities, which indicated the organization of those nanostructures within the myelin. So with the preferential organization along the phospholipid molecules. So another class of the carbon nanodots was the acetone-derived hydrophilic carbon polymeric dots. So they were very interesting because they were super cheap. You just need acetone and NaOH to synthesize it. The synthesis goes in 25 degrees, so there is no need for harsh condition, and we got hydrophilic carbon polymeric dots, also very small, although with higher distribution of sizes. And we also studied the inner composition depending on if we have the sodium or if we had the potassium as a starting molecules. The intensity was also nicely defined, although much wider than the previously described nanoparticles. The lifetime was also possible to be measured, and we've checked the two-photon excitation, which was around emission, which was around 1 micron for both types of nanoparticles. The emission was pretty strong, and for the 2-photon emission, we also saw its presence. The 2-photon absorption spectra were also wide, which is also good, in fact, because we can excite those molecules in a wide range of wavelengths with a pretty nice fluorescence. So we've chosen those nanoparticles to check the interaction with the proteins, and especially with albumins, because they are responsible for transport of many drugs. They have a competitive binding of calcium ions, the transport of hormones, and especially the ones which are fat soluble. They transport fatty acids to the liver and myocytes, and transport unconjugated bilirubin. So they are important in the sense of transporting. So we're wondering if the presence, if we would actually if we would like to image the living organism, if the nanoparticles would interfere with the main possible transportation way. So how the presence of the albumins will impact the fluorescence or in general activity of those nanoparticles. And it was very interesting because the fluorescence dyes are quenched by the presence of the biological media, and especially with the albumins, the major fluorescent dyes, fluorescence intensity decreases, whereas here we had a significant increase of the fluorescence in the presence of human serum albumin, and there was no emission of this albumin in this region, so it was definitely associated with the interactions of the carbon nanodots and the protein. We also define the Benesch-Hildebrand relation, which tell us how the carbon nanodots are associated with the proteins, and although they do interact, upon binding, we did not see the hampering of the function of those albumins. Moreover, the enhancement of the fluorescence was not only present upon 1-photon excitation, but also with the use of the 2-photon excitation. So in both cases, we've seen the significant increase of the fluorescence, which we also confirmed with the normalized 2-photon absorption spectra and the composites of the two. There was no efficient protein unfolding, as could be seen from the CD spectra. So the conclusion was that these carbon polymeric dots do not hamper functions of the globular proteins, so they could be safely used for the in vivo bioimaging. And last but not least, I would like to also introduce you to the folic acid-based carbon nanodots, where our precursor is folic acid, so a highly biocompatible molecule, which under ultrasound treatment and then with the hydrothermal synthesis and purification shows a great dispersibility in the aqueous media. So we reached 22.7 millimolar concentration with the precursor, which was only 3.6 micromolar. So the synthetic route was pretty efficient. We did have a just neutral aqueous condition of the synthesis, and it was performed in a stainless autoclave reactor, and purification was done on a column and thin layer chromatographies with the filtration process combined just with the water washing. So again, we do not use any harsh conditions for the synthesis, and as a result, we get quasi-spherical nano-objects of the average diameter estimated to be about 8 nanometers in this case. The chemical composition showed natural nitrogen doping and various bands responsible for the presence of various atoms. And in the internal design, we have the hydroxyl and amine moieties, the carbonyl groups, aliphatic chains, and some also aromatic domains. Those folic acid-based nanoparticles also show the excellent luminescent properties. Moreover, they were resistant to the pH in a wide range from 4 to 8 pH. There was intense blue emission of around 450 nanometers and a long-lasting fluorescence of 7 and 7.5 nanosecond. And as you can see here, the fluorescent quantum yield achieved in this case was over 50%. Again, we were able to excite those nanoparticles with 1 and 2 photon excitation, and we've seen the emission of strong emission in the first biological window. They are slightly blue shifted at the constant position, and they showed long-term photostability even under the femtosecond laser pulses. And the molar scale 2-photon absorption cross-section are comparable in the most fluorescent materials, inorganic, but they are mostly inorganic dispersions, for example, in toluene, whereas our carbon nanodots are actually dispersed in water. And again, we confirmed the possibility of marking of the myelin with those structures, both with 1 photon and with the multi-photon imaging. It's not well seen, but here's again the spoon-like structure. And also we've studied it within the 3D dimensions, so they were mostly present in the inner parts of the phospholipid. So to conclude, the various shapes of carbon nanodots may be achieved depending on the synthesis condition. Carbon dots seem to be promising alternative for other luminescent nanomaterials. Non-fluorescent folic acid molecules, when converted into ultra-pure folic acid carbon nanodots with excellent water dispersibility and superior optical activity. Non-linear optical properties in a wide range of wavelengths were measured and exploited for bioimaging, as well as I wanted to show you the myelin formation to various 3D structures depending on the external conditions, and myelin stability can be enhanced upon doping with the carbon nanodots. And here you can see how the formation of the myelin goes on. And I would like to thank especially Professor Lucia Petty for the invitation and the Polscom project overall and other collaborators, but most of all my group in Wrocław University of Science and Technology. Thank you.